everyone, welcome to my channel Pure Biology, and today I come with another video, that is the plant hormone abscisic acid. Okay, and before this topic I have already explained another biosynthesis of another plant hormone, that is auxin and ethylene. Obviously you just check out that videos. And before going to topic, I want to say thanks to all of you who have already subscribed my channel and support me for this channel. Okay, and obviously I try to give all the important information related to the biological science in future. So please continue with me and my channel. And today's topic is the abscisic acid. You can see here, this abscisic acid is the 15 carbon compound. Okay, this is the 15 carbon compound, and this abscisic acid generally remains in the plant's body in the cis form, not in the trans form. It produces sometimes a trans form, but it converts into the cis form. This is the sterilized form of the ADA. And this biosynthesis of the ADA, that is possible, or you can say that precursor of the ADA, that is the isopentenyl diphosphate, okay, or the IPP. Sometimes ADA that acts as the secondary metabolites, and you can check out my video that is related to the secondary metabolites, and there I already explained what is the secondary metabolites and what is the role actually in the plant body. So please you check out this video. And here the biosynthesis of the abscisic acid, how is that possible? The two regions, that is the plastin and the cytoplasm, both are very important region where the complete biosynthesis process of the ABA is occurred. Okay, so in that situation, first pyruvate and the glyceraldehyde 3-phosphate both added to form the IPP. Okay, isopentenyl diphosphate is formed. That is actual precursor of the abscisic acid. Clear? That is the actual precursor of the abscisic acid. And here the IPP that converts into the zeaxanthin, that is the C40 structure, zeaxanthin. Clear? This zeaxanthin transfer or converts, not transfer, it converts into trans-violaxanthin. Okay, trans-violaxanthin, that is C40. This trans-violaxanthin that converts to form the 9'-cis neoxanthin. Okay, 9'-cis neoxanthin, that is also C40. And here another enzyme, that is NCED, is one of the important enzymes that help to form the xanthol. Okay, xanthol from the 9'-cis neoxanthin. It converts actually 9'-cis neoxanthin to xanthol. Okay, and when it helps to produce, or it generally converts into the xanthol, these enter into the cytoplasm. Clear? Or you can say that after forming this, the total product that converts into the xanthol form, that enters into the cytoplasm. And here the xanthol that converts into the ABA aldehyde, that is C50. Okay, ABA aldehyde in presence of this enzyme SDR. What is SDR? Short chain dehydrogenase or the reductase. Short chain dehydrogenase or reductase. And after forming of ABA aldehyde, this converts into the abscisic acid in presence of abscisic aldehyde oxidase enzyme. Clear? Abscisic aldehyde oxidase enzyme. So here another thing occurring, that the stress-induced reaction. And what happened in that situation? The trans-neoxanthin that enters here. Okay, from this, in that site, another you can say that as a byproduct. The trans-neoxanthin that also enters here. Okay, so during the formation of the neoxanthin, from here, due to the stress, it enters in the form of the trans-neoxanthin added to this cis-neoxanthin. That means here the trans-neoxanthin and the cis-neoxanthin both are formed, but cis-neoxanthin is very important for the formation of the xanthol, and that enters into the cytoplasm. Okay. So this is the biosynthesis process of the abscisic acid. So abscisic acid here, what we know that, what we learn here, that abscisic acid is not only plant hormone, it sometimes acts as the secondary metabolites. Okay, it acts as the secondary metabolites. Now we learn what is the actual role of the abscisic acid. Okay, this is the biosynthesis process, and now we learn what is the actual role or the function of the ADA. So what are the functions of the ADA? First function, that is the dormancy and the germination. Actually, the seed germination or the dormancy, that is totally controlled by the ADA hormone. Okay. So how it is controlled by the ADA hormone? This is important fact obviously. How it is controlled by the ADA hormone? ADA hormone, actually, when the level of the ADA hormone is high, what happens? This inhibits the germination. Okay, this inhibits the germination, and you all know the seeds still cannot germinate. As such, they don't get the favorable condition. So they remain in the dormant situation, and this is possible due to the presence of ADA level or ADA hormone. Okay, so when the ADA hormone level is high, the seeds inhibit the germination. Okay, the germination of the seed is totally inhibited by the ADA hormone when the ADA hormone level is very high. But when the ADA hormone level is low, in that situation the seeds become germinated. Okay, so how is that possible? Actually, ADA that inhibit the gibberellin related, the hydrolytic enzyme. Okay, this is the synthesis of this hydrolytic enzyme that is related to the gibberellin synthesis. Clear? So this the enzyme is the alpha amylase, and what is the role of the alpha amylase? The role of this alpha amylase is it actually breaks the reserve food that is present in the seeds. Okay, it breaks the reserve food that present in the seeds, that is very important for the embryo that present within the seed, or when the seeds become germinated, and that situation the embryo gets their food by due to the synthesis of alpha amylase, and they get the food from the seeds, obviously, stored food or reserve food. So this totally stopped by the ADA level. So when the level of ADA that is very high, and that prevent the gibberellin acid or gibberellin hormone, you can say. So gibberellin hormone, and in that situation the gibberellin actually cannot help to synthesize the alpha amylase, and that is very important for the breakdown of the reserve food that present in the seeds. So that's why the seeds remain in the dormant situation. And this is the important factor that is controlled by the ADA. The second one is the vivifying. What is the vivifying? Vivifying, actually we already know here that the germination is totally controlled by the ADA. So in case of red mangrove seeds, due to the low ADA, generally they have the low ADA, and that is very important for them for getting the particular adaptations to grow in the soft mud, just below the parent tree. And we all know that where the actual tree or the parent tree remain, there after germination, the seedlings, they cannot grow there. Okay, so that's why the seedlings, they grow just far away from the parent tree. But in case of red mangrove seeds, they have the ability, or they gain the adaptive feature due to the low ADA. They can easily, the seedlings, they can grow in the soft mud just below the parent tree or near the parent tree. This is actual ability. Second one, this is the vivifying. And the third one is the hydraulic conductivity. What is the hydraulic conductivity? Hydraulic conductivity means the decreasing resistance to water movement across the membrane. Okay, this is the actually hydraulic conductivity. Okay, hydraulic conductivity actually the decreasing the resistance to water movement across the membrane. So this increases the HC or the hydraulic conductivity, that is ADA that increases the hydraulic conductivity of the plant's body. Okay, they can resist themselves. So, and ion flux of the roots in response to the water stress. So, they can easily survive in the water stress situation. Okay, they can easily survive. Plants can easily survive in the water stress situation. So, why it's possible? Because due to this ABA actually that increases the hydraulic conductivity of the plants, and that's why, and obviously the ion flux of the roots. Okay, ion flux of the roots. That means in the water stress situation, plants can get the iron or the nutrients from the soil through the roots. Okay, so this actually you can say this is one kind of adaptive feature that plants can gain themselves. And here obviously ABA hormone has important role. The last one, that is the roots and the shoots growth. ABA has obviously impact in the roots and the shoots growth. ABA generally, when ABA become very high, the endogenous hormone that exerts positive effect on the root growth, okay, by the suppressing the ethylene activity. So what is that? When the ABA become very high, in that situation the endogenous hormone that exerts actual positive effect on the roots to grow. And how is that possible? In that situation, the ethylene activity that is suppressed. Okay, so what happened? You can imagine that is vice versa. So ABA when the low, in that situation, it is the hormone that exerts the negative impact or negative effect on the root growth, because in that situation the ethylene activity is not suppressed or ethylene activity is high. And this situation generally happens when the water stress condition is occurring. Okay, so these are all the functions of the ABA. So today we learn about the biosynthesis of the ABA and the functions of the ABA. That is very important hormone, plant hormone obviously.